In this video, we're going to start studying some exceptions to Mendel's rules. So remember that Mendel said that in an organism you had two alleles that you inherited, and of those two alleles, one of them is dominant, one of them is recessive, so the dominant one takes over or is expressed, and the recessive one is hidden. And those are all principles that he discovered while he was studying pea plants. Now, genetics is not always that simple. There's a lot of exceptions to those rules, and if Mendel had encountered those exceptions, then his results might have been more difficult to interpret. So we're going to go over some of those exceptions today and do some problems involving those exceptions. Again, you should always review your pertinent terms, character versus trait, so please review those if you haven't already done so. And remember, we're talking primarily about inherited traits, which are traits that are passed on from generation to generation. So these are Mendel's assumptions. See, these are the assumptions that Mendel made, and it is under these assumptions that you'll usually get the ratios that we discussed. Now, we didn't discuss this 9:3:3:1 ratio. We will discuss that one later on. But we did discuss the 3:1 phenotypic ratio and the 1:1 genotypic ratio when you do a monohybrid cross. So if all these rules A to I are followed, then when you do a monohybrid cross with heterozygous individuals, you should get the 3:1 and 1:1 ratio. But sometimes we notice that those ratios don't happen, and it's because some of these little assumptions that Mendel made don't really happen. So in today's video, we're going to talk about two assumptions that don't always happen, especially in human genetics, but also in plant and other organisms. The first assumption is that alleles show complete dominance, and I'll go over when that rule is broken and when you see different cases of it. And then the second assumption, or let's call it a rule, is that there are only two alleles per character. So for example, for eye color, we said a brown and a blue allele, so there's only two alleles, big B or little b. For straight hair versus curly hair, you might have a curly hair allele and a straight hair allele, just big H and little h. But is it possible to maybe have more than one letter, more than two different types of alleles for a certain character? And so we're going to see that perhaps it is. So our goal today is to focus on A and B and see how that affects the ratios that we discussed in the monohybrid crosses. It would also affect them in the other cross called a dihybrid cross, which we haven't seen yet, but we will see it soon. So complete dominance is a degree of dominance. Dominance, there's different types of it. There's something called complete dominance, something called incomplete dominance, and something called codominance. Complete dominance is when one allele completely dominates over the other. It completely masks it. So for example, big G is dominant to little g. So when an organism has big G, little g, you wouldn't even know the organism has little g there, because based on the appearance, the organism is green. The only way to see little g is if little g is paired with another little g to make it yellow. So in this case, the dominant allele is completely dominant to the recessive allele, and the heterozygous individual has the same phenotype as the homozygous dominant individual. You can see here green and green, because big G completely dominates little g. That was the case with the plants that Mendel studied. However, complete dominance does not always occur. If you do have complete dominance and you do a heterozygous cross, you'll get your 3:1 ratio. For example, 3 green to 1 yellow. And then you'll also get your 1:1 genetic ratio, your 1 big G to 2 big G little g to 1 little g. So your 1:1 genotypic ratio, rather. Now, in some cases, you don't get complete dominance. You get something called incomplete dominance. In the case of incomplete dominance, the dominant allele is not completely dominant to the recessive one. It tries to overpower the recessive one, and I shouldn't speak that way, but I'm just simplifying it, but it doesn't. So instead what happens is you have an intermediate, a mix between the so-called dominant and the so-called recessive. If we have a red flower and a white flower, and red is incompletely dominant to the white, then we get a pink flower, an in-between color. Now when we talk about incomplete dominance, we sometimes use a different notation. For example, here I'm talking about flower color, so I'll use as my base letter C. And one of the alleles is red, like this. Here I'm talking about flower color, and that's why I'll use the base C, but the other allele is white, so I'll put a W on top. Now if I have two red alleles for flower color, the organism is red. If I have two white alleles for flower color, the organism is white. Now in incomplete dominance, the way I know I have that is once I cross these two individuals, CR, CR, and CW, CW, I get babies that are CW, CR, CW. These heterozygous individuals no longer have the same phenotype as the homozygous with the red. Instead, they have somewhere in between the red and the white. We have an intermediate, we call it. This would be pink. And that's how I know I have incomplete dominance. Now, if you make a Punnett square with the F1 generation, the heterozygous individuals, you have CR-CW on top, CR-CW on the side, you'll notice you get your red flower, two pink flowers, and one white. So phenotypically, this is no longer a 3-to-1 ratio. If it was dominant, we would have 3 reds, I'm just going to put R for red, to 1 white. If it was completely dominant. But because we have incomplete dominance, here's what we have now. We have 1 red, we have 2 pinks, and we have 1 white. Normally, the heterozygous individuals would all fall under this 3 category here, this 3 red here. But now that we have incomplete dominance, 2 of those individuals are heterozygous and therefore the intermediate, they're the pink. So our phenotypic ratio is no longer 3-to-1. Our genotypic ratio is still the same. We have homozygous of 1, another homozygous of 1, and 2 of the heterozygous. So we still have a 1-to-1 genotypic ratio, but our phenotypic ratio is no longer the same. So the heterozygous individual is no longer the same phenotype as the homozygous that we saw, for example, for red. It's an intermediate in between the two, which is why our phenotypic ratio is no longer the same. So when you do a cross with incomplete dominance, you can represent the alleles with a base letter and a superscript letter that represents maybe the color of that particular allele. You could also represent them using the normal notation. You could do big R, little r, but you have to remember that you have incomplete dominance. And you get a mixture, you get an intermediate between the two, which is pink. So again, you could use big R, little r. You could also do big R, W. But usually we like to use the same base letter if you're talking about a gene that controls the same thing. So that's why oftentimes in your textbook you're going to see C for color and then a specific color on the top. This way you know it's the gene that controls color, but this is the red allele specifically, and this is the white allele specifically in the case of incomplete dominance. So here we can see a summary of what we've seen so far. We have our 1-to-1 phenotypic ratio instead of our 3-to-1 phenotypic ratio. And you should be able to do Punnett squares with incomplete dominance. We'll do a few examples together later on. So in the next few slides, I have some examples with complete dominance, incomplete dominance, and something called codominance, which we'll see in a bit. When we talk about complete dominance, that's just the regular stuff you've seen before. So in your questions, when you're reading, you have to pay close attention to the degree of dominance that's stated in the question. So for example, here I'm stating that black feather color is completely dominant to white feather color. So if it's completely dominant and you have big B, then that means you know that the individual's phenotype is black. And if you have little b, then you know the individual's phenotype is white. And since it's completely dominant, you know the heterozygous individual is going to be the same phenotype as the homozygous dominant individuals. So this is just like the regular questions that you've seen before. Determine the genotypes of the following phenotypes. So if you have a black chicken, you know it's going to be big B, little b, or big B, big B. A white chicken, you know it's little b. And this part here is kind of a trick question. Because it's complete dominance, you won't have a genotype that will give you a gray chicken. You can then do your regular crosses like you would normally and come up with your phenotypic and genotypic ratios. On the next slide, you're going to see a similar question, except this time the black feather color will be incompletely dominant to white feather color. So it's incomplete dominance on the next slide. So you can see that over here, we have incomplete dominance. So you read that question carefully. Based on that information, you should be able to come up with the phenotypes, genotypes, and do some Punnett squares. So for example, if I have CB, color of feathers, black allele, this should be black. And if I have CW, white allele for feather color, then this should be white. But now in the case of incomplete dominance, if I have a heterozygous individual, the B and the W here, that should be some mixture between the two colors. And if you mix black and white, then you get a gray color. You should then be able to find the genotypes for cases of incomplete dominance. So for example, for a black chicken, I would expect the genotype to be C big B, C big B. And it doesn't really matter what if you choose uppercase or lowercase for incomplete dominance. Now, if you had put CB, CW, that would have been incorrect because, again, in this case, the B is not dominant to the W. It's incompletely dominant. So that would actually not be a black chicken. That would, in fact, be a gray chicken, CB, CW. And the only way to have a white chicken is to have the homozygous with the white alleles for feather color. And so you should be able to do Punnett squares with incomplete dominance. I'll do one example with you. I'll do example F over here with you. So we have two gray chickens. Make a Punnett square and describe the phenotypic and genotypic ratios. So for my two gray chickens, I know they'll be CB, CW crossed with another CB, CW. And so what I'm going to do now is I'm going to put the alleles in the Punnett square like you would normally do for any Punnett square. I'll put the father on the top here, or the mother, it doesn't really matter, they're both the same genotype. So CB gametes, CW gametes, so the gametes are made in the same way, CB or CW. And then over here, same idea, we have a CB or a CW. And then you simply match them in the squares. So here we have CB, CB. Here we have CB, CW, CB, CW, then CW, CW. And so instead of having that 3-to-1 ratio that you would normally see in the complete dominance, we have 1 black, 2 grays, 1 white. So we have a 1-to-1 phenotypic ratio, not 3 blacks to 1 white. This is no longer the case because of incomplete dominance. And so it's the same idea. To make gametes, it's the same idea as well. So if you have CB, CW, an individual would have either the option of making CB gametes, 50% chance, or CW gametes, 50% chance. Here we have some more practice problems for you to attempt. So go through the PowerPoint, do try these out. Make sure you read the questions carefully. Pay attention to that idea of incompletely dominant instead of completely dominant, and whenever you see that, it just results in a intermediate between the two colors that you have. So read the questions carefully, answer all the questions, see what's being asked in the question, and show all your work. There is one more degree of dominance, something called codominance. I like to refer to codominance as no dominance, because in this case, both alleles are dominant. They both appear in the heterozygous individual. It's not going to be an intermediate. It's not going to be a red and white make pink. It's going to be that you're going to see both, for example, red and white together. So you're going to get a red/white. So you're not going to get a pink. You're going to get a red and white together. You're going to see some red parts and you're going to see some white parts. So let's take a look at this example here. Here we have a red bull, and here we have a white cow. And the H is the base color for the hair color. And we have little r, little r. And again, it doesn't really matter if you put big R, little w, big W, because you don't really have a dominant allele. They're codominant. Whereas in complete dominance, you'd put a big letter to show that the big one is bigger and takes over in the phenotype. But in that case, it doesn't matter here. So I'm going to have HR, that's going to be my red bull. That's the gametes that the red bull will produce. And then I'm going to have HW. Those are the gametes that my white cow will produce. And then we do the fertilization events in the Punnett square. And you'll notice you get HW, all HW. Now, if this was complete dominance, if red was completely dominant to white, then they would all be red calves, but they're not. What happens is you actually see some red fur and some white fur both showing up. So it's not a pink calf, it's actually some red and white both showing up. And so that is what we call codominance, where they both dominate. They both kind of show up and express in the organism. And then if you were to do a cross between these F1 generation calves, you would get not a 3-to-1 ratio anymore, but rather a 1-to-1 ratio. You'd get 1 red to 2 roan. Roan essentially means red and white. So I'm just going to call that R/W, and 1 white. So it's very similar to the 1-to-1 phenotypic ratio that you got in the incomplete dominance. It's just that rather than being an intermediate between the two, you actually see both expressed within the heterozygous individual. So again, rather than have a 3-to-1 phenotypic ratio, rather than have 3 red to 1 white, 2 of the reds that would normally be red, the heterozygous ones, are the intermediates. And then 1 stays red as long as it's homozygous with the 2 red alleles. So again, same idea. You should be able to answer questions relating to codominance. So look for that keyword in there as well. So you might see complete dominant, completely dominant, you might see codominant, and you might see incompletely dominant. And that will affect the phenotype of that heterozygous individual. It's the same idea in terms of the alleles and the gametes that can be formed with the genotypes. We'll do just a couple examples together here. So if we have CB, you know that that would be a black feathered chicken. If you have CWCW, then you know that would be a white feathered chicken. And if you have the heterozygous and we told you it's co-dominant, then you know it's black and white. The name we give for that type of chicken is an erminette chicken. Black and white. That's the name we give for the black and white feathered chicken. So if you had to write down the genotypes for the phenotypes, so a black chicken, in the case of co-dominant, has to be CB. And again, it could be a lowercase b. It doesn't really matter if it's uppercase or lowercase in these scenarios. For a white chicken, CW. And for the black and white chicken, you make sure it has two different alleles, CB, CW. And you should be able to make crosses with Punnett square crosses. So, for example, over here, I'm telling you that you cross two erminette chickens with each other. And so if you do a Punnett square with that, you should be able to predict what the phenotypic and genotypic ratios are. So we'll do that example together. We know the erminette chickens are the heterozygous ones. They have to be if they're co-dominant. So CB, CW crossed with CB, CW. Put your alleles on top and the side. So CB, CW, CB, CW. And do the fertilization events. So CB, CB. There's our first black chicken that we get. CBCW. There's our erminette chicken. CBCW. There's another erminette chicken. And then CWCW, a white chicken. So rather than get 3-to-1, again, our phenotypic ratio is a bit off this time because we have some co-dominance occurring. So we can see we have one black, two erminettes, and one white. Not a 3-to-1 ratio like you're used to seeing. So now we're going to take a look at a quick animation to see what dominance really means. How does it relate to genes? How does it relate to stuff in the body? So again, your genes are codes in your DNA. And you can see the genes on the chromosomes. Here we have a gene for, let's say, red flower color. And on the other chromosome, the homologous pair, you have a gene for white flower color. So suppose that these had a completely dominant relationship. Suppose red was completely dominant to white. Here's what that would mean. The red version of the gene codes for a red protein. And the white version doesn't code for anything at all. No pigment protein. Pigment's like a protein that paints stuff. So that red version would code for a red protein, and that would go to the flower. And you just have red protein being added to the flower, and a lot of red protein because it's completely dominant. Let's just say the base of the flower is white, and you're just adding some red paint to it, and you have so much that the flower ends up being completely red. So you don't see any white there at all. The only way you could have a white flower is if this one here was also white and there was no red protein added to it. So that would be an example of completely dominant. Let's take a look what this would look like if it was incomplete or co-dominant. So for the case of incomplete dominance, it would be similar to the complete dominance case, except our red gene here, we just don't have enough red protein being made. So we're still producing some red pigment, but we kind of have to spread it thin over the white base, and as a result, we get an intermediate between the two. We get some pink instead. So again, what we're seeing is that the dominance is really based on the interaction of the gene product. What that means is what is being made by that gene and what is it doing to the organism. So this gene here, this allele for red, is producing red pigment, but in the case of incomplete dominance, there's just not enough red pigment to completely color the whole thing red, so we spread it thin over the white base. It's a bit of a simplification, but It's the general idea. Now let's say there was codominance. If we actually had codominance happening, and we had two different alleles, well, what that means is that both the alleles produce protein. The white one produces white pigment, the red one produces red pigment, and they both color the flower together. And then we get codominance. We get some white showing up and some red showing up as well. So at the genetic level, you'll learn more about that in grade 12. That's what explains codominance, complete dominance, and incomplete dominance. So again, just make sure you review what dominance actually means. Dominance just means which allele is expressed in an organism. Just because something is dominant, it doesn't mean it's the more common one or the most popular one or the better one for a population. In fact, there are many diseases that are dominant. So for example, if you had big R, little r, that would mean you have a disease. Let's just make up a disease here. So you have a disease if you have big R, little r. And that big R allele may not be very common, because what would happen is the individuals who do have that disease maybe didn't have time to reproduce and pass that gene on. So what's more common in the population is actually the recessive one. So it all really depends on what the allele does to you that determines whether it's more common or not if it's dominant or recessive. This last point here you can see says natural selection determines the frequency of an allele, which basically means if the allele is not good for the population and the individuals can't reproduce because of that allele, it's going to be wiped out. So if a dominant allele is bad for a population, over time the individuals of the dominant allele will be wiped out and your dominant allele number would go down, whereas your recessive allele number would go up. So dominance has nothing really to do with how frequent it is in the population. It's more so how it expresses itself in an individual. So here we have some examples of different types of dominance based on some descriptions. And you'll see it's not always clear cut to see if something is completely dominant or incompletely dominant in an individual. Sometimes it depends at what level you're looking at it. And so what you're going to do is you're going to read through these descriptions here, and you're going to figure out if you're dealing with a case of complete dominance, just put C, codominance, put CO, and incomplete dominance, just put I, based on the description provided. So pause the video and then come back and take a look to see how you're doing here. So the first one, Tay-Sachs disease at the organism level. It says that to have Tay-Sachs, you need to have both recessive genes. If the individual does not have both, the individual will not have Tay-Sachs. So what that means is there is a clear difference between the heterozygous individual and the recessive individual. So hopefully you chose that this means that this is complete dominance. Now let's go and zoom in at the biochemical level, just a different level of studying the organism. It says that the activity level of the lipid metabolizing enzyme in heterozygotes is intermediate between that of in individuals that are homozygous for the normal allele. So if you look carefully at the biochemical level, you have an intermediate between the homozygous, your heterozygous is an intermediate between the two extremes. So if you're an intermediate, that means you have incomplete dominance, intermediate, somewhere in between. Now if you look at the molecular level, even zoomed in more, you'll see that heterozygous individuals produce equal number of normal and dysfunctional enzyme molecules. So you're expressing both the good enzyme and the bad enzyme. So both alleles are being expressed, and hopefully that you saw that that was codominant. Now we're not going to be dealing with a lot of that, but I just want you to get that depending on what level you study, something might be codominant, something might be incomplete, something might be complete dominance. But I'm not going to go out and trick you. You should be able to solve regular problems with this and hopefully understand this general concept. Here are some more sample problems that you can attempt with regards to the different types of dominance. So make sure you try those out. Here there's a summary of the complete and incomplete and codominance that we studied in this PowerPoint. And there are also some practice problems along with some answer keys, and there are some other practice problems that will be provided to you in class. This concludes the first rule that we saw. Mendel studied complete dominance, but in reality there's also incomplete and codominance that exist, and that kind of throws off our phenotypic ratios. Rather than having 3:1, we have 1:1 and 1:1 because of that new phenotype that's created by that intermediate or by that codominance effect that we saw earlier. In the next part of this PowerPoint, we're going to take a look at what happens when you have more than 2 alleles. So rather than just having black and white allele, what if you have 3 alleles? How will that affect the organism? Here you can see a summary of the different types of dominance. Again, single dominance just means complete dominance. Number of alleles, we have 2, so all of the types of dominance have 2 alleles. We just represent them in different ways. So for example, here I might have big B and little b, whereas here I might have CB and CW. The genotypes that are possible, you have 3 possible genotypes for all of them. Remember, you have the homozygous BB like this, or little b like this, and the heterozygous like this. But for incomplete and codominance, it's the same idea, except you represent them with this little format here: CBB, CWW, and then of course our heterozygous individual CB, CW. Where things really look different is in our phenotype. For complete dominance or single dominance, you only have 2 phenotypes. You have, for example, the black, which could be big B, or big B, little b, and the white, which is little b, little b. However, when you have incomplete dominance and codominance, you now have 3 possible phenotypes. One of them is that blend that was created, the intermediate, and the other one, in the case of codominance, is the case where both of them appear. And the new phenotype really is in that individual that's the heterozygous individual, so the CB, CW. So that was when things don't show complete dominance, we might have different ratios. Rather than 3:1, we see our 1:1 phenotypic ratio. Another assumption that Mendel had is that there's only 2 alleles. So for flower color, there's only big R and little r, or just CR and CW. But in some cases, there are some characteristics that have more than 2 alleles, and we're going to study one particular case in humans called blood types. So here we have the different blood types that can exist in humans. We have type A, B, AB, and O. And the type of blood that you have determines your ability to donate blood to other people or to receive blood from other people. Now, blood type is genetically determined. Based on your genotype, your blood cells will look different from somebody else's genotype, somebody else that might have a different genotype. So if you're, let's say, IA, so I would be the base letter, and A would be the superscript. If you have IAIA, like this, or IA little I, your blood cells will have this little carbohydrate on it, and that triangle carbohydrate is what makes your blood type A. Now if you have IB, or IB little I, your red blood cell is going to have this little circle on it, that little circle carbohydrate on it, and that's going to be a type B blood cell. Now, if you have, let's say, IA and IB, both of them together as your genotype, then you're going to have a little bit of both. You're going to have the circle and the triangle, and that'll give you type AB. And if you have little I, then you won't have any carbohydrates on the membranes of your red blood cell, and you'll be type O. So you may have noticed that there's more than two alleles. It's not just allele A and B, you also have little I. So you have allele IA, IB, and little I. IA codes for the triangle protein, or carbohydrate rather, and IB codes for the circle carbohydrate. Now in reality it's actually coding for an enzyme that adds the carbohydrate. An enzyme is a type of protein. So IB would code for an enzyme that adds B carbohydrate, and then IA would code for an enzyme that adds triangle carbohydrate or A carbohydrate. And then I would not code for any. So we have multiple alleles. We have 3 alleles instead of just A and B. So because we have 3 alleles, we have more than just 3 types of genotypes. We have IAIA, IAI, IBIB, IBI, IAIB, and little I, little I. So we have 1, 2, 3, 4, 5, 6 possible genotypes for blood types. And as a result of having more genotypes, we also have more phenotypes. We have 1, 2, 3, 4 phenotypes. So in this case, your genotype is which allele you contain, which 2 alleles you contain, and your phenotype is which blood group you're part of. I mean, we could describe which carbohydrates we have in your blood cells, so we don't really see that directly, and it's harder to measure. So what we do is we just determine with some reactions and some tests what blood group you're part of. So one more thing I want to point out before we do some practice problems is that you may have noticed that IA is completely dominant to I. When you have IA and I, you become blood type A. The only way to become blood type O is if you have 2 little i's. So the little I here is recessive to IA. And you'll notice that IB is also completely dominant to little I. So there's 2 cases of complete dominance. So in blood typing, you have complete dominance, but at the same time you may have noticed that you also have codominance. So IA and IB, they are both expressed at the same time on the red blood cell, and therefore you're a combination. You're an AB blood type. So you have IA, IB, that's codominance. So your blood type is really an example of having multiple alleles, but it's also an example of complete dominance and codominance at the same time. The big idea I want you to get is that you have more alleles, you have more genotypes, and therefore more phenotypes that are possible. And the alleles, those genes, determine which enzymes you make, which determine which carbohydrates you have on your red blood cells. Now, you can do Punnett squares with these genotypes and phenotypes just like you did with the previous examples in the same way. The genotype is written in the same way as the sort of incomplete and codominance, and the gametes work in the same way as well. So for example, if I have an individual who's blood type A and we say is heterozygous for blood type A, that means they're IAI. If they're homozygous for blood type A, that means they're IA. This individual here could only make IA gametes because there's only IA alleles. This individual here could make IA gametes, 50% of them, or little I gametes, 50% of them. So it's all the same concept, just a slightly different variation of that theme. So here we have a summary of what I explained before. You have more genotypes because you have more alleles, and you can see the possible gametes that can be made. And then we have some practice problems over here that you can attempt in order to see if you understand the concept summarized in this table here. So I'll do a few problems with you. You can probably do some on your own already. So you can pause the video, try as many as you can, and then compare your answers. I won't do all of them, but I will do some of them with you. So a man has blood type O. What are the possible genotypes? Hopefully you put little I, little I. A man has blood type A. What are the possible genotypes? Well, you can go and look in your table, or you might already know that to be A, you could be homozygous A, IA, or heterozygous A, IA, I. If you're AB, that's the example of codominance, you have to be IA, IB. If you're blood type B, same idea, IBI or IBIB. A child is blood type O, what are the possible genotypes? Same idea, little I, little I. So hopefully that's enough training to know how to correlate the phenotype to the genotype, and then you should be able to do Punnett squares. So I'll do one example with you. A woman with blood type AB is crossed with a man with blood type O. What are the possible blood types that your children can have? So if you have a woman with blood type AB, let's write down her genotype. So I'm going to do it up here in this corner. We have IA, IB, crossed with an individual that's blood type O, little I, little I. You can make a Punnett square much the same way you've done it for any other question. So let's make our square here. We have IA, IB on top as the gametes for the AB person. And we have little I on the side here as the gametes for the type O person. And then you can do the fertilization event. So we have IA, IA, IB, I, and then IB, I. So the only possibilities of kids would be A and B. So 50% of the kids would be blood type A, 50% of the kids would be blood type B. If you have a kid that's blood type AB, then that means that you had a mix-up in a hospital, and that's a possibility. So this is a great way to see if the kid that you have actually belongs to you, or was maybe a mix-up in the hospital. So you should be able to do many practice problems now that relate to blood types and Punnett squares. Attempt the rest of these ones here. Compare answers with each other to make sure that you're on the right track. You'd write the ratios in much the same way. If I were to ask you for a phenotypic ratio for the Punnett square we just did, you'd say 1 type A for every 1 type B. If I were to ask you for a genotypic ratio, you'd say 1 IA, a genotypic ratio, 1 IA for every 1 IAI for every 1 IBI. So where do blood types come into play? Well, there are some people called phlebotomists. They are technicians that are trained to draw blood, and oftentimes if you want to donate blood or receive blood, you need to know your blood type. So there are some blood types that can receive blood from anyone. They're called the universal donor. And then another blood type is called the universal receiver. It can receive blood to anyone. So universal donor can give to anyone. Universal receiver can receive from anyone. So you can take a look at this table here and probably figure out who the universal receiver is and who the universal donor is. So if you look at blood type A, you can see that they can receive from A or O. And if you look at blood type B, that person can receive from B or O. So you know that A and B are most likely not the universal receivers. But let's look at blood type O. Blood type O can only receive from O, so that's not a universal receiver. But blood type AB can receive from everyone. So because blood type AB can receive from everyone, that would be the universal receiver. How do we know who the universal donor is? We'll take a look at who can at which individual shows up in every spot in this table here. So A can receive from O, B can receive from O, AB can receive from O, O can receive from O. So O is the universal donor. And it all has to do with your immune responses. Remember that, let's say you're a type A individual, that means you have those triangles on your red blood cells. Now, let's say you then get some type B blood added into you, you're suddenly getting some red blood cells with little circles on it. And your body is not used to seeing those circles. So what will happen is your body will create what's called antibodies to attack those invading circles that it's not used to seeing. And then it'll cause your blood to clump up, and that's called agglutination. Now you've probably guessed based on this example that blood type O can be given to a blood type A because O doesn't have those carbohydrates on it, so your immune system isn't going to freak out. Likewise, if I was a blood type B, so this way instead, and I was giving blood type A to the blood type B person, the person with blood type B is used to seeing circles, but when they see triangles, they freak out. If you're a blood type AB, well, you're used to seeing everything. So if you're AB, you have triangles and circles. It doesn't matter what you throw into the body, you're used to seeing the triangles and circles, and you won't panic and create antibodies to cause your blood to clump together or agglutinate. So you can see the agglutination process that I was talking about on the previous slide in this table here. So if you are blood type O, you're going to produce these anti-A and anti-B antibodies, meaning their defense against the A carbohydrates and the B carbohydrates, because you're not used to seeing those. So let's say you're given blood type O, your blood will look just fine. But if you're given blood type A, now your blood's going to clump up. If you're given blood type B, it's going to clump up, or AB, it's going to clump up. So O can really only receive from another O. If you look at someone with blood type A, they have the anti-B antibody, which means they're not used to seeing Bs. They're going to have defense against the B, that circle carbohydrate. So if you give it some O, it's fine because O doesn't have anything on it. If you give it some A, it's fine because A is just like itself. But if you give it some B and you have those circles on it, now it's going to start clumping together. You can see all these little red blood cells clumping, and same thing with the AB. Yes, there's A on it, but there's also B on it. And the same idea applies to blood type B individuals. And then if you look at blood type AB, well, they don't really have any antibodies. They're used to seeing everything. They have triangle and circle, so you won't see any clumping at all when you add different blood types to it. So this AB here is definitely a universal receiver, and this O is definitely a universal donor, because you'll notice that it doesn't trigger any responses, agglutination responses. And AB does not have any agglutination responses regardless of what blood type is put into it. So here you have some practice problems for blood types, codominance, and incomplete dominance. There will be a research assignment that you'll have to do to learn about the Rh factor, the rhesus factor. You may have heard of A positive, B negative, B positive in the blood type, and this is just another protein that is genetically controlled. So that's something that you will have to research. There will be a guided worksheet that you'll work on. So you don't really need to do number 10, because the guided worksheet will help you to complete number 10 as you work through it. Here are some great questions for you to attempt. It's a good problem-solving one. You should compare your answers with a partner to see if you were on the right track. You basically have three parents with their blood types that are known, and there are three babies, and they were mixed up in the hospital, so you have to figure out which baby belongs to which parent based on the blood type. Again, here's a summary of the different blood types that we've seen based on the genotypes, what the blood type is, what antibodies they produce. The antigen is the carbohydrate that's present on the red blood cell itself. The antibody is sort of that defense molecule that triggers that agglutination. And you can see who you can donate to and who you can receive from. And then you can also see your universal donor versus your universal receiver over here. Recipient is the same thing as a receiver. A receiver is the person who gets something, is the recipient of something. So again, you will have the opportunity to study what the negative and positive mean. It all refers to this protein called the Rh protein. If you do have the Rh, your O positive or A positive or B positive or AB positive with the Rh. If you don't have the Rh on your cell, red blood cell, then you are negative, 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 and negative. So it's just a way to let you know if you have that Rh protein or not. This does have implications for pregnancy, and when you do the research sheet, you'll see what that implication is. The idea is that sometimes the mother may not be compatible with the baby. So you can see here the mother, genetically speaking, is an Rh negative mother in her blood, and the baby is an Rh positive in the blood. And so what could happen is if some of this positive goes out into the mother's blood, the mother is going to start producing some Rh antibodies, some defense proteins to get rid of the Rh that now came into her blood. And if that Rh antibody goes into the baby, it's going to start attacking the baby's red blood cells. And if you don't have red blood cells, it's going to be very, very difficult for the baby to carry oxygen to itself, and it could get anemia or get very, very, very sick. And sometimes you might have to deliver the baby early if it's very severe. So you'll learn that there are ways to prevent that. You'll learn about when this is more likely to happen, and what this process is called. It's called the Rh factor sensitization. There's a little video here that explains the concept, and there will be a PDF reading that goes along with it to make sure you understand the idea. This little infographic shows you how you can prevent that whole Rh sensitization. So please make sure that you watch the video and read the infographic in order to make little notes about how you can prevent it. In your textbook, the readings that correspond to these concepts are in pages 194 to 196, and then you should have enough information to be able to do questions 1 to 10. There will be other worksheets provided, and there will be the Rh research sheet that you should also work on.